Hello, my name is Selvi Arliani, and in this video I will talk about propagation of microalgae.
The important things that we need in order to start a microalgae propagation are culture media, concentrated microalgae culture, nanochloropsis species, seawater, improved Neubauer hemocytometer, pipette, conical flask, light microscope, Bunsen burner, measuring cylinder, test tubes, and tally counter. Next, for the procedure, all apparatus was sterilized. The mouth opening of the conical flask containing nanochloropsis species was sterilized by flaming using Bunsen burner. 90 ml of nanochloropsis species were taken out and were added into a new conical flask containing Walnis medium. The mouth opening of the conical flask were covered by cotton wool to prevent contamination. The solution mixture was then placed in a live food culture laboratory with the presence of light source at 23 degrees Celsius. On the next day, cell counting were being carried out by using Neubauer hemocytometer. 1 ml sample of nanochloropsis species were being taken out by using micropipette twice from the conical flask and was labeled as test tube 1 and test tube 2. The average cell reading and cell density was being calculated. After 10 to 14 days under this condition, green algae will appear in each flask. If this color not appear after this period, repeat all procedure above. After 2 weeks, check samples with a microscope for contamination by taking 1 drop of the sample to the microscope slit and cover it using cover slit and check under power lens 40 times if there is any other species in the culture. After 2 weeks, transfer algae from each 1 liter flask to separate 5 liter flasks that containing 4 liter of sterilized and fertilized seawater. Keep these flasks covered and aerated under laboratory condition. Next, prepare the bag that's suspended to a rack. Fill the bag with sterilized water and leave enough free space for the volume to be inoculated. Wait a couple of hours to check for possible leaks. If found, seal them or replace the bag. Next, introduce nutrients and inoculum. Fit 2 PVC tubes for aeration to the bottom of the bag. Connect them to an air line and adjust the airflow to a gentle bubbling. Add the nutrients using a graduated cylinder at the usual rate of 1 milliliter per liter of each working solution. Select a mature culture that will be used as inoculum, either from a large flask or from another bag. In case if a flask is used as inoculum, remove the caps and flame its neck. Then, close with a flame aluminum foil stopper and let it cool. After that, remove the stopper and pour the algae culture from the flask into the bag. The inoculum should be about 10% of the receiving volume. On each bag, mark that algal species, origin of the inoculum, and the bag serial number. Algae culture can be attained cell density of 10 times 10 power of 6 cells per milliliter within 5 to 7 days. Daily routine works includes determining the algal density and monitoring algal water temperature 1 time a day at 7 a.m. When the algae reach the stationary phase, it is better to harvest them before they going to dead phase. Bioflocculation method is recommended to harvest the microalgae because It enables the harvesting of microalgae without addition of chemical flocculants and allows for the use of the cultivation medium without any additional treatment. Another advantage of this method in comparison with other applied flocculating microorganisms is that it doesn't require different cultivation conditions and therefore avoids additional costs and prevents undesired contamination. The presence of the flocculating microalgae in the final biomass concentrate does not interfere with further downstream processing of the lipids into biodiesel. The algal biomass can be used as biofuels, animal feeds, aquaculture feeds, food supplements, nutraceuticals, and pharmaceuticals. That's all from me. Thank you.